So you are probably pretty used to the idea of AI writing things for you by now, right?
Oh, yeah, absolutely. I mean, it drafts emails, it generates poems, it can even write, like, complex Python scripts in seconds.
Right, it's just everywhere. But consider what happens when the text the AI is generating isn't made of English words or computer code. Like what happens when it's writing in the language of DNA?
That is where things get really crazy.
Yeah, and taking it a massive step further, what happens when scientists take that, computer-generated code, literally print it into physical existence in a lab, and then watch it come alive?
I mean, it honestly feels like something pulled straight out of a hard sci-fi novel.
Sure.
But this is exactly what is happening in biotechnology right now. We are crossing this historic milestone where the boundary between a digital algorithm and physical biological life is becoming, well, highly porous.
Okay, before you start picturing like an AI printing out a T-Rex or hallucinating some apocalyptic supervillain pathogen, we really need to establish the sandbox we're playing in today.
Yes, very important clarification.
Right, because our focus is entirely on bacteriophages,
Yeah.
or just phages, as they're commonly called. And these are viruses that exclusively infect bacteria, right?
Exactly. They do not have the biological hardware to infect humans, animals, or plants. So they basically offer researchers a relatively safe, microscopic test bed to see what generative AI is truly capable of when applied to living organisms.
Which brings us to the core of what we are unpacking in this deep dive. We're looking at a stack of fresh research, primarily a landmark paper from the Arc Institute, alongside a very sobering commentary published in Science.
Right, and the objective here isn't just to marvel at the fact that AI can generate a virus on a screen.
No, we need to look at the mechanical reality of how these models learn the hidden grammar of biology, like how that digital sequence is physically synthesized into a functioning biological entity, and what this paradigm shift means for both personalized medicine and global biosecurity.
Yeah, so to understand how an AI can write a functional virus, we really have to look at how it learned to read biology in the first place.
Right, because you already know that DNA is built on those four base pairs, A, T, C, and G. But imagine feeding millions of those completely unannotated genetic sequences into a massive neural network.
It's exactly like how a large language model processes human text, right?
Mhm.
It ingests massive data sets to learn statistical relationships between words.
Exactly. Like it doesn't actually know what an apple is. It just calculates that the word apple is highly probable to follow the phrase red and juicy.
Right, right. And genome language models operate on that exact same underlying principle, but applied to molecular biology.
Which is wild.
It is. One of our sources highlights an early milestone in this specific field, which is the mega DNA model. The researchers trained this AI on roughly 100,000 bacteriophage genomes.
And the critical detail here is that these genomes were completely unannotated, right?
Yes, completely. No human scientist went into the data and flagged, this sequence is a structural gene or this is a promoter region. The AI was fed nothing but raw, endless strings of nucleotide bases.
There's a continuous sea of letters.
Exactly, but the megaDNA model utilized a multi-scale transformer architecture, so it wasn't just reading the sequence linearly, one base pair at a time.
Oh, I see. He was utilizing attention mechanisms across multiple layers of scale.
Yeah, exactly. It would analyze tiny windows of just 16 base pairs to catch local, immediate motifs, like a ribosome binding site, for example, and simultaneously zoom all the way out to evaluate global topologies up to 96,000 base pairs long.
Wow. So by processing the data at multiple resolutions like that, the model naturally began to map the structural syntax of life.
It did. And the researchers devised a brilliant way to test if the AI actually understood the biological weight of what it was reading.
Oh, how so?
They prompted the AI to predict which genes were essential for a virus's survival, and it performed this in a zero-shot manner.
Wait, zero shot, meaning it had never been trained on what a lethal mutation looks like.
Exactly, nor had it been explicitly taught the very concept of an essential gene.
So, if the AI evaluates a sequence by mathematically measuring its loss, which is basically its error rate or its statistical level of surprise, how does that translate to biological viability?
Well, if it expects a specific sequence based on the vast grammar it has learned, and you digitally mutate those letters, the model's loss function spikes.
Oh, I get it. It's the algorithm's way of sensing a lethal mutation without needing a biologist to label it as such.
Yes. When the researchers simulated mutations in genes that are absolutely vital for the virus to replicate, the AI's loss spiked massively. It recognized that altering those specific localized sequences would destroy the coherence of the organism.
And it's just mind-blowing. And beyond just reading and evaluating, it could generate entirely new sequences up to 96,000 base pairs long, right?
Yeah, and these generated sequences featured highly realistic regulatory elements like promoters to initiate transcription, all positioned exactly where they should be mathematically. To a computer, it looked flawlessly like a real virus.
Okay, I have to push back here for a second, or at least admit some genuine confusion about this leap. Because generating data on a screen is one thing.
Yeah.
Like a language model can write a blueprint for a car, drawing the wheels and the engine, and it looks mathematically perfect on your monitor. But biology is, wet, messy, and totally unpredictable.
Oh, absolutely. It's highly chaotic.
Right. So a text prediction model doesn't inherently understand how complex proteins fold in 3 dimensions or how they interact with cellular thermodynamics. How did a model predicting the next token make something that actually breathes and functions in a petri dish?
That is the fundamental gap between computational biology and physical virology. I mean, a blueprint is useless if the building collapses the moment you lay the bricks.
Exactly.
So to bridge that gap, the researchers at the Arc Institute moved beyond megaDNA and utilized incredibly powerful frontier models called EVO1 and EVO2.
Okay, and how are these different?
Yeah. Well, these models were pre-trained on a staggering 2.7 million phage genomes. And to get them to design something specific and testable, they fine-tuned the models on a data set of about 15,000 genomes from a specific viral family called Microviridae.
Ah, and they gave the AI a highly specific biological template to work from, right?
Yeah.
The FIX174 virus,
You got it.
which, for anyone following the history of genetics, is famous for being the very first DNA-based genome ever fully sequenced back in 1977. It's this tiny, elegant workhorse of a virus, packing only 11 genes into about 5,400 base pairs.
Right, it's very compact. So the researchers prompt the AI to generate new viruses that share the general architecture of FIX174, but with entirely novel genetic sequences.
And what happened?
The AI generated over 300 novel designs, and this is where the physical laboratory comes in. They didn't just leave these designs on a server. The researchers engaged in actual physical synthesis.
Meaning they took the digital sequences and ordered actual synthetic DNA.
Yes. Through a very meticulous process, they assembled these synthetic genetic fragments into full circular viral genomes, and then they introduced this synthetic DNA into living E. coli bacteria.
Using laboratory techniques that essentially force the bacterial membrane to take up the foreign genetic material.
Exactly.
I mean, this is the ultimate stress test for an AI hallucination. If the AI's multi-scale grammar was off-like, if it made a single fatal error in structural dependency among those 5,400 base pairs, the DNA would just sit inside the bacteria completely inert.
But it didn't. Astonishingly, 16 of these completely synthetic bacteriophages worked.
Wait, really? 16 of them?
Yeah. They successfully hijacked the E. coli cells, co-opted the bacterial machinery to replicate themselves, and then lysed or burst open the bacteria to release the next generation of synthetic phages.
Oh my God. The digital code successfully crossed the barrier into physical functioning biology. That is profound.
It really is.
I mean, we have used CRISPR to edit existing genomes, but this isn't just fixing a few typos in a natural genetic sequence. This is asking an AI to draft entirely new chapters from scratch, and the biological machinery actually reads it and executes it perfectly.
It's a completely new paradigm.
But I do have to ask about the novelty here.
Yeah.
How different are these 16 functioning viruses from their natural FIS174 template? Are we just looking at minor iterative tweaks, or are they genuinely distinct?
No, they are far from minor tweaks, and analyzing them leads to the absolute jaw-dropper of the study. Let's isolate one specific AI-generated phage, which they cataloged as EVO536.
Okay, EVO536, what's special about it?
Well, viruses utilize a highly specific structural protein known as gene J in this family that is essential for packaging their DNA into their outer protein shell, or capsid. In EVO536, the AI completely swapped out the natural gene J for one belonging to a completely different, evolutionarily distant virus called phage G4.
Wait, you can't just drop an alien component into a highly optimized biological machine.
No, normally you can't.
It's like trying to put a modern graphics card into a computer motherboard from 1998. You can't just jam it in, the connecting ports won't align. The hardware fundamentally doesn't know how to interface.
Right, and human scientists have actually tried that exact experiment. Prior to this AI research, biologists manually attempted to take the gene J from G4 and insert it into FIX174 in the lab.
And let me guess, it failed.
It was a complete failure. The resulting virus was dead because the structural dependencies didn't match. But the AI succeeded where human engineering failed because it didn't just copy and paste the gene, it inherently understood the structural context.
So to make the new graphics card work, the AI completely rewrote the connecting ports on the motherboard.
Exactly. It subtly co-adapted the intergenic regions, which are the non-coding sequences between the genes, so that the alien protein could interface perfectly with the existing viral shell.
That's insane. It mapped the structural dependencies and accommodated the new parts simultaneously. That implies the model has an implicit understanding of the three-dimensional protein folding we were just talking about, entirely derived from recognizing patterns in one-dimensional text sequences.
Yeah, it is a remarkable emergent property of the transformer architecture operating at scale. Now, we must maintain scientific accuracy here.
Fair enough, let's ground it.
The AI did not conjure life out of a vacuum. It relied on known biological templates, and it required immense computational filtering to weed out the vast majority of designs that likely wouldn't fold correctly.
Right, and it required rigorous physical lab testing. Out of over 300 designs, only 16 were viable.
Exactly. It is an iterative, heavily filtered process. But the achievement of synthesizing structurally novel functional biology that solves complex molecular engineering problems remains undeniably groundbreaking.
Absolutely. And, if an AI can reliably write viable, novel viruses, the immediate application that comes to mind is the escalating global crisis of antibiotic resistance.
Oh, without a doubt.
Because when traditional antibiotics fail against superbugs, clinicians sometimes turn to phage therapy, right? You use natural viruses to hunt down and kill the resistant bacteria.
Right, that's the standard approach.
But if the natural virus is locked out because the bacteria mutated its surface receptors, how does printing a batch of AI-designed phages solve the problem? Don't they just hit that exact same evolutionary wall?
Well, natural evolution is a bottleneck. When you unleash the natural Fix174 virus on a target, it's highly effective initially. However, the bacteria rapidly evolve countermeasures.
Right, they adapt.
Yeah, they change the molecular structure of their outer surface, specifically their lipopolysaccharide synthesis. They essentially alter the molecular docking ports that the virus uses to attach and inject its DNA. And once those receptors change, the natural virus is totally useless.
So they just lock the door. And the Arc Institute researchers set up a confrontation to test this, didn't they?
They did. They took 3 highly resistant strains of E. coli designated CR1, CR2, and CR3. These strains had completely altered their surface receptors, locking out the natural Fix174 virus.
Okay, so the natural virus is useless here. What do they do next?
The researchers then unleashed a synthetic cocktail consisting of their 16 AI-generated phages onto these resistant superbugs.
What happened?
The synthetic cocktail rapidly overcame the bacterial resistance, and the mechanism by which it happened is just fascinating.
Let's hear it.
As the AI phages infected the bacterial cultures over a few generations, they engaged in horizontal gene transfer. Because multiple different synthetic phages were infecting the same bacterial cells simultaneously, their replicating DNA strands broke and recombined.
Oh, wow.
Yes, specifically, modular genetic chunks from EvoF111, EvoF114, and EvoF147 swapped material to form entirely new, ultimate recombinant phages that completely decimated the resistant bacteria.
That is wild. The AI essentially injected an artificial reservoir of genetic diversity into the cocktail. Diversity that natural evolution just hadn't generated for this specific phage. Exactly. And that massive toolkit allowed them to rapidly recombine and out-evolve the bacteria's defense mechanisms on the fly.
Furthermore, some of these AI designs were fundamentally vastly superior to their natural counterparts in terms of raw viral kinetics.
Wait, really? Better than nature?
Yeah, in head-to-head growth competitions, one AI-designed virus called EvoFi69 vastly outcompeted the natural wild-type virus. Another variant, EvoFi2483, was significantly faster and mechanically stronger at lysing the host bacteria.
So they were highly optimized biological machines engineered for that specific bacterial environment.
They really were.
The clinical implications of this are just staggering. I mean, we are looking at the foundational architecture for programmable, custom-tailored phage therapies.
It changes everything.
If you're battling a multi-drug-resistant infection, doctors might not have to scour environmental samples, hoping to find a naturally occurring phage that happens to match. We can utilize an AI to search through an impossibly vast landscape of biological possibilities, sequences natural evolution never happened, to test and physically print a custom-designed cure on demand.
The potential to revolutionize personalized medicine and combat the superbug crisis really cannot be overstated here, but the ability to computationally design completely novel biological entities introduces an unprecedented asymmetric risk profile.
Right, so we've essentially built the ultimate biological skeleton key. But if this AI can pick any lock, what happens when it designs a key for a door we explicitly do not want opened?
That is the big question, and we need to quantify just how alien some of these AI creations are to natural biology. Take one of the functional generated phages, EVO5 2147. When researchers aligned its genome against its closest naturally occurring relative, they found 392 mutations.
In a genome that is only about 5,400 base pairs long, 392 mutations is a massive divergence.
It is huge. It shared less than 95% of its average nucleotide identity with any known phage in existing databases. And in the taxonomic classification of virology, falling below that 95% identity threshold essentially classifies the organism as a brand new species.
Wait, so the AI basically hallucinated a functional, novel species of virus?
That's exactly what happened, yeah.
Okay, so in the cybersecurity world, there's a concept known as a zero-day exploit. It's a piece of malicious software that targets a software vulnerability that no one knows about yet.
Right.
And the reason 0 days are so devastating is that traditional antivirus systems rely on recognizing the signature of known threats. If the malware is entirely novel, the security system simply lets it walk right through the front door.
And that is exactly what we are facing here. We are looking at the biological equivalent of a 0 day.
Because global biosecurity screening systems are built on homology, right?
Mhm.
They scan synthetic DNA orders to see if they match the genetic sequences of known pathogens.
Yes, and so how does a screening algorithm flag a brand new, completely synthetic threat generated by a transformer model? It can't.
Wow.
It represents a severe blind spot in our current biosecurity infrastructure, and it is the central focus of that commentary piece published in Science.
Did the ARC Institute researchers see this coming?
Oh, they are acutely aware of this dual-use dilemma, and they built robust safeguards into their methodology.
Okay, what kind of safeguards?
When they were training the EVO1 and EVO2 models, they utilized a technique called data exclusion. They deliberately scrubbed all eukaryotic viruses from the training data set.
Oh, meaning the models were never exposed to the genetic sequences of viruses that infect humans, animals, or plants?
Precisely.
It's like teaching an AI the deep structural grammar of French, but deliberately withholding every single word of Russian.
That's a great analogy.
Like, even if you prompt it to, it cannot suddenly write a novel in Russian because those specific lexical connections just do not exist in its weights. It literally lacks the biological vocabulary to build a human pathogen.
Right, that is the intended safeguard. However, the Science commentary highlights a critical vulnerability in relying on data exclusion.
Which is?
The foundation models themselves, these multi-scale transformer architectures, are becoming democratized. The governance required to safely steer this technology globally just does not yet exist.
I see where this is going.
Yeah. A bad actor, whether a state-sponsored entity or an independent rogue group, could utilize cloud computing to take a foundational genomic model and fine-tune it on eukaryotic pathogen data themselves. They could bypass the data exclusion safeguard entirely.
They could just upload the Russian vocabulary themselves.
Exactly that. And the commentary points out an even more chilling reality regarding the democratization of the physical synthesis process.
Because generating the dangerous DNA sequence is only half the battle, right? You still have to physically assemble it in a wet lab.
Exactly. But studies cited in the commentary have demonstrated that current large language models are already capable of acting as highly sophisticated lab assistants.
Right, really?
Yeah. These models can coach individuals step by step through complex virology protocols, troubleshooting assembly errors, and outperforming up to 94% of PhD-level virologists in experimental planning.
That is terrifying. So the barrier to entry isn't just dropping, it's practically disappearing. You no longer need a decade of specialized postgraduate training in molecular biology to manipulate a viral genome.
Not anymore, no.
You just need, access to an open source model, a benchtop DNA synthesizer, and an LLM to walk you through the wet lab protocols.
It is the ultimate double-edged sword. The exact same technology that gives us the power to eradicate untreatable superbugs simultaneously lowers the drawbridge for novel biological threats.
Right. It forces us to look at the grand trajectory of where biotechnology is actually heading.
Yeah, and the consensus among the sources is that the debate is no longer about whether generative genome design will become a reality.
Because the threshold has been crossed.
Exactly. The computational models work, the physical synthesis pipelines function, and the resulting synthetic biology is viable. The defining challenge of the next decade is how society will capture these revolutionary medical benefits while our regulatory frameworks and biosecurity screening mechanisms race to catch up.
It's just a perpetual race between our capacity to invent and our capacity to protect.
It really is. And the final thought I want to leave you with stems directly from the long-term implications discussed by these researchers.
Let's hear it.
We are rapidly approaching an inflection point where generative genome design becomes a standard, scalable tool in biotechnology. Today, the focus is on small, relatively simple bacteriophages. But as computational power and context windows expand, what is the next logical step?
Right. Where does it end?
We are looking at a future where we could generate entirely synthetic, complex living organisms from scratch. Imagine querying an AI to design a bacteria perfectly customized to degrade microplastics in ocean water. Or, an organism engineered to survive in extreme industrial environments to synthesize clean fuels and then simply printing it into existence.
I mean, the implications are almost too vast to comprehend. We may be entering an era in which code does not only run on computers, it can sometimes become biology. If you enjoyed this story and want more surprising science explained without the homework, please like the video and subscribe for more.